Test. 1, 2, 3, test. Okay, it's working. All right, so today we're going to talk about inducible expression in algae. This is our first biotech paper that we're going to discuss. After we were so rudely interrupted. Okay. This is the first paper we're going to discuss. This is Jackson et al. 2019. Okay, so I'll just kind of briefly introduce some stuff, and then as we go through, I'll ask questions, and I hope that you guys will participate with the question and answers. Okay, so this is Nannochloropsis. This is a single-celled algae. It's a couple things about it. It's haploid. It's eukaryotic. Okay. So this is not an archaean. It's eukaryotic haploid. It's marine. So what does that mean? It means it lives in the salt water. So like salt water. And the nice thing about this system is that it has a single chloroplast. So if you look at, here's a diagram of this cell. There's a single chloroplast. And so an algae like this, like Chlamydomonas and Nannochloropsis, are becoming models for photosynthesis, at least in a single-celled eukaryotic sense. So I'm starting to get into Nannochloropsis because I'm interested in photosynthesis, and photosynthesis obviously underlies the basis of all agriculture. So I'm starting to get into this as like a model organism, and it's becoming more of a tractable model, and that's precisely what this paper is about. It's essentially trying to build out systems that make this a better model for biotechnology and for studies. Okay. Other things about Nannochloropsis. So this was on the quiz. What is Nannochloropsis useful for? Yes. So Nannochloropsis, so the answer was biofuels, in case for people watching this later. Nannochloropsis produces these lipids, these fatty acids, which can be converted into biodiesels. So people are interested in trying to figure out how to get good cultures of this and potentially genetically modify it to get better production of biodiesels. Okay. Other things about this is that maybe this was on the quiz. This is a system that's genetically tractable. What does that mean? Take a guess or help me. It does have a genome. Yes, that contributes to making it genetically tractable. What else? Yes, it can be transformed. That's actually kind of a big deal. If a single cell can be transformed with a plasmid, it makes it much more easy to manipulate. So not only can it be transformed, but there's also papers where people are now using CRISPR-Cas9 systems to modify this. They are able to do integrations onto the genome. Okay. So it's an interesting system for biotechnology and photosynthesis in general. Okay. Now let's ask, what is the purpose of this paper? Which is pretty much like essentially anytime whose paper is this their first paper that they've ever read? Hopefully nobody. Okay, so people have read like scientific papers before. So a good title of a scientific paper should essentially tell you exactly what the purpose of the paper is. So what's the purpose of this paper? Yes, so they want to build a plasmid that is inducible, that they can turn on and off and control it. And essentially they're testing this nitrate reductase promoter. Okay, that's the simple gist of this paper. And I'm curious, so after we got through this first test, when you guys tried to read this paper, did the words now make sense to you? You're able to understand it? Okay, then I feel like I've done my job. So that's why the first test is always kind of hard. It's a lot of learning the language. But if you were successfully able to read the text of this paper and understand what was happening, then like I did my job, and now you guys are able or should be able to actually read the primary scientific literature. Okay, so if they're gonna build this plasmid that will be able to express things in Nannochloropsis, what is the plan to do that? So first give me like a broad, like what's the basic design of the plasmid in a broad sense? Okay, so there should be, okay, so in general, the plasmid, the selectable marker, the promoter. We know that the promoter, which is described in the title that they're discussing, is the nitrate reductase promoter. Nitrate reductase promoter. So we'll call this NRP. And they're going to build this promoter. What's the selectable marker that they're going to use in this system? That was on the quiz, I think. Beta. Started with an H. Yeah. Hygromycin B. Hygromycin B. Okay, I don't need you to memorize hygromycin B, but now you should have the framework of understanding that every organism is a little bit different. But if it can accept a plasmid, you'll probably be able to find a selectable marker for that organism. Okay, and it happens that hygromycin B is a selectable marker that works pretty well in Nannochloropsis. Okay, so in general, this would be the basic design of the plasmid, but what do they want to actually express? Yes. Okay. So the actual gene that they would, in theory, clone in is this EGFP. So it's essentially GFP, except it's enhanced. So you can make a few modifications, a few mutations in GFP, and it will fluoresce better. So that's what EGFP is. What's the purpose of putting GFP in this plasmid? Yeah. So essentially, like, we said that biotechnologically you would want to build these plasmids, and in theory you'd be able to express like any protein you wanted. But in practicality, to first begin experimenting in the system, you usually do something that's very simple, like EGFP, so that it has a simple readout. Like you'll be able to see whether it's working or not. And then the idea is if it's working, then we can take out the EGFP and we can put in something else that might actually be useful, might actually do something. But for now, GFP doesn't really have a purpose per se. The purpose is to get the system working and to have a readable marker that will tell or determine if the system is actually working. Okay. I'm debating what to go into next. Okay. Let's describe the process to build this construct. This was also a question on the quiz. So in simple terms, what's the process to build this plasmid? And actually, there's a few things we're missing. There's still something else that we're missing. If you read the methods, they put in a terminator. So there's a terminator sequence that they clone in. Is there anything else? So they actually, yeah, okay, I'm just looking at the notes. They actually clone a promoter, a gene, a terminator, etc. So there's multiple pieces. So what's the strategy to build the plasmid? Who was able to read the method successfully and understand the strategy? It kind of was like a mix of things that I did teach and something that you should be able to extrapolate. So here's the actual paragraph. So okay, so what was said was injected in yeast and run a PCR. So let me just correct the language a little bit. You wouldn't inject into yeast, you transform into the yeast. So the one part of what you said is correct. Is yes, they use yeast. Okay, they're using yeast to build the plasmid, and then you said, what did you say after that? They inject into yeast, and then what? And you said run a PCR. So it's some kind of a weird combination of PCR and using yeast, right? Hopefully everybody understood that. Can anybody elaborate that in a more accurate, more precise way of how they're actually going to build this plasmid? Yes. Okay. So that's correct. So they run PCR, or it's actually various multiple PCR reactions, and then they use homologous recombination in the yeast to actually assemble the plasmid. Okay. So This relies on a technique that has a term. It has a name. It's called gap repair. So gap repair is probably like worth explaining. Gap repair is just a form of homologous recombination. So we all should know what homologous recombination is. And we remember particularly that yeast is particularly good at homologous recombination. You don't need any fancy plasmids or any fancy stimuli. Yeast just does homologous recombination very well. Okay, so within that subset of homologous recombination techniques, there's a technique called gap repair. Gap repair is when you have, say, a plasmid, okay, and you want to insert something onto that plasmid. What you can do is you can take a homology region. Let's call this homology region 1. Let's call this homology region 2. Okay, homology region 1, homology region 2. You can clone those homology regions through PCR. So maybe clone isn't the correct term, but you can PCR amplify any sequence X, flanked by those homology regions. And then to actually initiate gap repair, what you do is you linearize the plasmid. So you do a cut between those regions. Okay. So you linearize the plasmid. Linearize the plasmid just means you cut it, you open it up. Now you've produced something that looks like this, except these homology regions should be purple. Okay, so you've linearized the plasmid. Now let me ask a question. Are linear plasmids stable in yeast? What happens to linearized fragments in most cells? They get destroyed by cellular exonucleases, okay? So linear DNA fragments are typically like unstable, especially plasmids. So this is not necessarily a stable plasmid. But encoded on this backbone is a selectable marker also for the yeast. Anybody catch what that was? I think they use for, if you saw in the reading, there's a plasmid called PRS416, something like that. I think that is a Uro3 marker. So on this backbone is a yeast selectable oxytrophic marker, okay? And if you combine, so this fragment, let me grab this quick. If you combine these two elements, so this 1 plus 2, into yeast, so you put these both in a budding yeast cell, and you put it and you select for it, or you put it under growth conditions, which is minus ura dropout media. What's the yeast gonna have to do in order to survive? Well, let me just clarify your language. You're right, but let me clarify your language. So what was said was pick up the plasmid. The problem is that the plasmid is not circular and it's unstable. So what will the yeast be forced to do? It'll be forced to induce recombination to fix the plasmid, to repair the plasmid. So you see, that's why this is called gap repair. You can cut out segments or linearize the plasmid. And then as long as you pair the plasmid with a receptor or a secondary piece of DNA that could combine with it. So what would happen is let's actually draw this out in a spot where we have space. So here's part 1. Assume that like at a molecular level, these things align and you can get a recombination and a recombination, and this produces a circularized plasmid. Okay, so the yeast has the power to close up the plasmid, and you will select for that when you use the dropout media. This technique is called gap repair. Okay, so gap repair is a cloning technique that involves linearizing a plasmid, mixing it with something that's going to recombine with it, putting it into yeast, selecting for the recombination, and then you'll get the recombination. So now to just finally like clarify and elaborate the very precise methodology here, what they're doing is they're PCRing sections that are going to get cloned into this plasmid, and then they're causing gap repair in the plasmid and yeast and selecting for these recombinants. And the actual section where they describe this is right here. Vectors were generated in vivo by homologous recombination in yeast to obtain the native promoter sequence of the nitrate reductase gene. This is talking about how they're cloning or producing the amplicon. A region of 1.3 kb upstream the gene was amplified using these primers, blah, 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 blah. So they design primers for this promoter right here, the nitrate reductase promoter, from the endogenous sequence. So where are they actually pulling, or what is the template source of DNA for the nitrate reductase gene? Yes, the genome of what? Yes. So they purify genomic DNA from the algae. Somewhere in that gene is the nitrate reductase gene, and in front of that gene is 1.3 kb bases of sequence that encodes the promoter for the nitrate reductase gene. And they design primers for that, and they put their homology regions on those primers, and then they can clone that out and do their gap repair, okay? Let's see if anything else is worth reading here. Let's talk about the GFP sequence. So there, you can see that they're cloning this sort of like stepwise, piece by piece. And it says here that the GFP sequence was amplified from P gap alpha GFP using the GFP primer pair. So then my question then is, where does the template DNA for the GFP gene come from? Comes from this thing, whatever this thing is. What is this thing? Well, okay, so let me contrast this with this. In the case of the nitrate reductase promoter, they clone the nitrate reductase promoter from algae genomic DNA. What I'm trying to point out here is, did they clone the GFP gene from algae genomic DNA? What was the template? Well, so that's what I'm trying to clarify is, so historically, yes, GFP gene was in a jellyfish, but are they cloning that gene from a jellyfish here? Where's it coming from? It's coming from this thing, which is what? Yes. Okay, that's what I was looking for. So the reason you know that this is a plasmid, how do you know that this is a plasmid? Yes, this is how you know that this is a plasmid. So again, like, you're starting to understand the language. Like, when I read these things, like, would I expect that you know what P gap alpha GFP is? No, I don't even know what that is. Like, all I know is that that's a plasmid. And the reason I know it's a plasmid is because it says that little P right in front of it. It's P gap alpha GFP. It's a plasmid, and on that plasmid somewhere is this GFP gene. So essentially what's different from cloning the promoter is that in the, sorry, this thing is like not letting me scroll to the right and left. What's different from the cloning the promoter is that they have a template plasmid, and on that template plasmid is some EGFP, and they probably just pulled out this plasmid from their freezer because they had this. So they pulled it out from one of their libraries, they designed primers, okay, that are going to amplify that gene, and they probably put their homology regions, and then they did another gap repair reaction to insert the EGFP. Okay, the point of this is now you should start to understand and see that to assemble these plasmids often requires like multiple steps, and where you get the DNA is not always the same. Sometimes you're taking genomic DNA. If you're working with something like a marker that is included in many different plasmids, maybe you will pull that from a mini prep from a library plasmid that you already have. So you could use plasmid as a template to clone something. You could use genomic DNA as a template to clone something, etc. Okay, so that's how they get those two pieces. Using GFP primer pair, blah, blah, blah. The native ATP synthase alpha subunit terminator was generated. So in this case, when you see the word native, when they're talking about the terminator, that means that the terminator sequence was cloned from the endogenous algae DNA, just like the promoter was, okay? So now you're starting to understand how to read these methodologies. What's important to include in the methodology is how you're cloning it, what you're cloning, where the source of the DNA comes from, either the plasmid or the genomic DNA, and then how you do that, like potentially listing out your primers, etc. So we should all kind of be able to understand this. I'm looking at this other stuff. The hygromycin gene was amplified from another plasmid. So the hygromycin cassette came from a plasmid. So it's essentially like a mosaic where they're pulling different pieces from different things that they have, and they're combining it into this plasmid. So that's the strategy of how they actually build this plasmid. Okay. Good question. Yes, typically if you're reading a molecular biology paper, plasmids will always have the little prefix. So you see something like this, that's typically means like that's a plasmid. Good question. Okay, so here's the map of the thing that they built. It's pretty complicated. This is the hygromycin cassette. They put this promoter. It's probably worth talking about this promoter. This promoter is described right here, UEP, the ubiquitin extension promoter protein promoter, UEP. What do you think is the properties of this promoter PUEP in front of this hygromycin cassette? Well, I more so, I mean, well, it's not an enhancer, it's a promoter. But what? Yes, it's always on, it's constitutive, and would you say it's strongly expressed or weakly expressed? Very strongly expressed. And the reason you know it's strongly expressed, like this is sort of more advanced biochemistry knowledge, but ubiquitin is a very, very strongly expressed protein. And so if you know that this is a very, very strong constitutive promoter. So the strategy is to put a very strong constitutive promoter in front of the selectable marker so that you'll get good selection, okay? Now, on the other hand, they're not doing that with the GFP. With the nitrate reductase promoter, how would you describe that promoter? Yes, it's an inducible promoter. So how do you induce it? You add nitrate. I should know this. What's the chemical formula for nitrate? Anybody know? I'll just write nitrate. NO3. Okay, so if you add nitrate, the promoter turns on. There's another thing that is interesting about this promoter. It's not only just inducible, it's also repressible. How do you repress it? Yeah, you add ammonia. So hopefully NH4. Hopefully I'm correct about that. So if you add ammonia, it will repress it. So this should give you very, very tight control of this GFP, okay? And then as we discussed, there's a Euro 3 cassette, because what they did is they gap repaired it into the backbone of this PRS 426, which is a yeast plasmid. It's got the 2-micron origin, so it's a high copy in the yeast. In E. coli, it's got this PBR 322 origin, which is a high copy origin, and it's got an AMP selectable marker for E. coli. So this plasmid has like a ton of stuff on it. It's compatible with yeast, it's compatible with E. coli, and now what they built is it's now compatible with algae. So it's like a 3-way compatible plasmid. Okay, once they built this, how was the plasmid verified? Did anybody catch that? So in the methods, there's a section, molecular cloning. That's where all this stuff is. Yes, very good. It was restriction digest. I'm looking where that section is. Here it is. So it says, so there's a little line here that says, the assembled plasmid was transferred from yeast to Escherichia coli, and following restriction digest screening, the cassette was released from the backbone by digestion from PME before transformation into the algae. So essentially this is the only line that tells you how they validated the sequence. And all they're saying that they did is they did a restriction enzyme digest and they cut something out, okay? So let's ask some questions about that. Would you say that that's a good strategy or a bad strategy? Yes. Okay, that's a good answer. So the answer, what was said was it's a good starting point. You're right that that's a good starting point. Like my thought is to build this plasmid and to produce this complicated plasmid map probably required multiple PCR reactions and multiple building steps. And so to just after you've built all that at the very end, do one restriction enzyme digest that cuts out one thing is pretty bad analysis. Like that's a start. What they should really do is they should honestly sequence this entire plasmid. They should do Sanger sequencing, or nowadays you can kind of like do like a it's like cost like $15 to sequence an entire plasmid. They should honestly have done that. Or at the very, very minimum, they should have done multiple restriction enzymes instead of just one, and they should have checked a few like different restriction digests. So that's one sort of criticism of this paper is it's not really sufficient to build something this complicated and then just assume everything is correct and do like one restriction digest. So that's kind of, it could be done better. So that's just a little criticism there. But that's how they validated that. Okay. Okay, so the next question. How was the, wait, let's look at the figures. So here's figure 2. What's going on in this figure? So let's first just dissect A. What's A? They have these different numbers. These are, okay, these are green cultures. What are these cultures? D algae. Why are they green? This is an interesting question. Are they green because of the GFP, or are they green because of their algae? In this case, like algae are always going to be green because they have chloroplasts, right? So there is kind of this interesting conundrum, which is the creatures are already green. So what? So that's a good question. So GFP is fluorescent. What does fluorescence actually mean? Like when you say it's fluorescent, what does that actually mean? How do you fluorescence physically means what? It's not just about reflection. So you're right that fluorescence means like there's some kind of an excitation of electrons. They do their orbital thing and then they're releasing photons at some certain like energy, right? So that is definitely fluorescence. But to get fluorescence, you have to add something. Like fluorescence sort of implies that you are exciting the electrons by shooting like lasers at it, essentially. Not necessarily lasers, but when you induce fluorescence, you usually have like a little apparatus that will shine light onto the, in this case, the algae. And that light is at a very specific wavelength that causes the GFP to fluoresce. So my point is that to the naked eye, I'm sorry, this is kind of like nuanced. You can kind of, with the naked eye, see some fluorescence. Like if you make fluorescent E. coli with GFP, you can see green E. coli. But it's not really fluorescing until you shine a light at the right excitation wavelength. It's like a black light. Like when you go into, yeah, it's exactly like a black light, where when you go into like a music store or whatever, and then there's a black light room, and then you see all this stuff shining, that's what real fluorescence is. So in this case, what you're not, the reason it's green is not because of the GFP. The reason it's green is because of the algae. Okay. That's just sort of a side point. Well, that's a good question. That perhaps that could be a few different things. Let's make some hypotheses. Why do you think some of these could have different color of green? Some have what? Yeah, it could be a cell density. So it could be just that some of these clones, these are different clones, these numbers, those are different clones. Some of these clones perhaps grow faster. Perhaps they grow to a higher concentration. What could be some other hypotheses for why you see some different greens? It could be what I just said it was not. It could also be like maybe you're seeing a little bit of GFP. Who knows? Yeah, like maybe. It could be the GFP. After I just said like, no, it's not that. It could be. But the question is like you don't know. Like you don't know, and I wouldn't trust it. Like I don't know. I don't know why some of these are darker, some of these are not. Okay, but these are the clones. Let's just make sure we didn't miss anything. So they're definitely not, I would never trust that. They're definitely not, at least I'm not. I did not look at this data and think, damn, 8, that one's real dark. That one's got high GFP. Did that correlate? No, that doesn't correlate. So if you look at, so here's clone 8, you might think this clone is the darkest green. Therefore, look at the expression. Here's expression of GFP in clone 8. That's definitely not one of the higher ones. So that proves my point that the darker, after I said no, then yes, then now I'm saying no again. The darker one is not expressing higher GFP. Okay, let's look at B. What's going on here? What is this? What type of data is this? Okay, it is a gel. I don't want to give away the answer. You can look at it, you can look at B and you are able to tell just by looking at B whether it's an STS page or an agarose gel. What do you need to look for and what do you need to see? Yes, very good. Okay, so you see the kilobases. Because you see the kilobases, you know right away that this is DNA, this is nucleic acid, therefore it is an agarose gel. Yeah, pDNA. Okay, so this is like a control where they're putting some plasmid DNA, I'm assuming. Okay, so now what's actually happening here? What are they running on the gel? When you say the clones, that's not specific enough. What are they actually running? Like they're not taking algae cells and running algae cells on the agarose gel. This is not a restriction digest. So this is not, yeah, this is not a restriction digest of plasmid. And how do you know that? Look at the legend. What key word in the legend tells you that this is not a restriction digest? Right, so figure legends are sectioned into part A. Here's part A, which is describing this thing. And then there's part B, which starts right here and goes up here, which is describing this thing. So what's the key words that tell you what these things come from? Right, it's in this paragraph. Well, I'm thinking this. I mean, genotyping by PCR using so this is a PCR reaction. That means that these are amplicons. Okay, so they've got some gene that they're PCRing where they've got primers, and the amplicon produced is precisely right around 1 kb. Okay, that's how you know, is they tell you right there in the legend. And the gene that they're PCRing is the hygromycin. So these are the different clones, and they're running a PCR for hygromycin. So what is actually the conclusion that? What is this actually telling you? It's telling you that when you have these clones, you do a PCR reaction of their DNA and you get a positive band for hygromycin. What's that confirm? Yes, they have the plasmid. The only thing that this figure tells you is we have successfully transformed these and they have the plasmid. Okay, that's really the only thing that this tells you. Here's another question. Why don't they just do a mini prep? Like you've heard and I've taught that you can just do mini preps and purify plasmid. Why don't they do that here? Mini prep is not too much work. So the answer was maybe it's too much work. Well, in this case, they have to do a bunch of genomic DNA extractions for every clone, and they have to do a bunch of PCRs. So if you could just run a mini prep, it'd probably be a faster pipeline. But you guys are not catching the real reason why they can't just do a mini prep. You're real close. It's not that it's a eukaryote, but it's no longer in E. coli. Like you can do mini preps in E. coli because you can grow E. coli and LB at real high density. And then there's a very defined protocol for pulling out plasmid from E. coli that gives a very high yield. And this plasmid in E. coli, if you look at the backbone, this plasmid in E. coli is going to be very high yield. But what's going to be the copy number when it's actually in the algae? Do we even have any idea? No, we actually don't even really have any idea because they're building kind of like the first systems for the plasmid. So I don't even know if this is a high copy or a low copy. It could be super low copy plasmid in algae. So the point here is you can't just do a mini prep of algae cells with a plasmid that you have no idea what the copy number is. That's kind of like a E. coli specific protocol. So in this case, if you don't know whether it's high abundance or not, you run something like a PCR, because if it's there but it's at low abundance, you'll definitely see it with the PCR because it's exponential. Okay, so that's why they do this PCR reaction. Are there any questions about that? Okay, so they've essentially just proved that they can do these transformations. Okay, let's look at this figure. So here's figure 3. Let's look at, what do we want to look at here? My first question is, why is there so much variability? Like what you're seeing here on these graphs, the peak of the green bars is some measure of the intensity. Okay, so in this case, let's actually just look at the y-axis. So in A, what is actually being measured here? Yes, the nanograms, so the amount of protein per cell. So this is actually protein per cell. Okay, this is a different y-axis. What is this? Fold change. This is I think this is essentially testing induction, isn't this? Yes, that's my read on this, is that B is actually measuring the fold change of induction of the protein when you add the ammonia or the nitrate. Okay, what? So that's my read on this. So these are essentially like two different kind of, but similar methodologies to quantify the amount of protein they have. And on the x-axis, you have the different clones. So one thing that's for certain is that the clones are not expressing the GFP in a consistent manner, strain to strain. Okay, so if you compare different strains, they have very different levels of expression. What would be some reasons for that? Let's generate some hypotheses, and they actually do talk about this, I think, in the discussion. What are some hypotheses for this variability? So they said that. They in the discussion, they do say that. They say it's possible that there are integrations into the actual genome. Okay, and they actually use the word positional effects. So they say it's possible that this variability is caused by positional effects when a plasmid integrates into the genome. What is the counter to that? So first of all, they don't know. They don't actually know. They're hypothesizing and they're discussing this in the discussion. What's a counter to this idea that perhaps there's these integrations and they have these positional effects?
No matter where it's at on the plasmid, if you think
Yeah, but okay, so you're kind of close in that, or you're close to my thought in that you said the word plasmid. Let me follow that up with a question of how, what makes them think there are integrations? Were they trying to do an integration? An integration is when you have the chromosome of the algae and you build a plasmid, and then that plasmid recombines onto the chromosome, and now you have this thing integrated into the chromosome. Did they actually do that or intend to do that? Well, they built the plasmid with gap repair in yeast. were they intending to do chromosomal integrations? They were not intending to do that. At least I didn't catch that in my reading of it. What I understood is they're just trying to transform the plasmid and hope it hangs around in the algae and see what the effect is. They didn't actually build a system of integration to get actual efficient integrations. So I think they're like that's a hypothesis. Sometimes you do randomly get just random plasmid integrations, and they're essentially saying like maybe that's the reason. Maybe it's integrating. But they were not trying to do that. So what makes them think that that actually happened? I think there's other plausible scenarios that can generate this variation. What's another scenario that can generate this variation? That could be different. So you're saying there could be like epigenetic background thing. So epigenetics is when the protein modifications on the DNA actually change the expression of the DNA. That could be true. That's a decent hypothesis. It could be that. Except these are all on plasmids. So I doubt that they'd be targeted for like epigenetic modification, but it's possible. That's definitely a possibility. What else? What else could cause this variability? There's something that's super simple with just plasmid basics that I'm looking for. Well, they're gonna evenly distribute the nitrate in the media of which the algae are eating. So the nitrate should be consistent throughout the mixture. Yes, you're getting so what was said is the origin of replication. What you're really hinting at is the copy number. We have no idea what the copy number strain to strain is, and there's no reason, according to this plasmid map, there's no programming within algae that dictates the copy number. Okay, perhaps we don't even. I don't know enough about algae to know algae specific origins and how you encode those. But according to this map, I don't see an algae specific origin. And so it's highly probable that all this variability is just due to differences in copy number, strain to strain. Okay. The other thing to think about with this paper or with this figure is this fold change measurement. The fold change measurement is important to counteract something called autofluorescence. What's autofluorescence? Say that again. Yes, when it glows without you even doing anything. So you can take almost anything, like any organism or any object, and you can put it in a microscope and you can shine a fluorescent exciter at it, and you'll see some green because you're like shining things that cause like green fluorescence. That's called autofluorescence. So the idea is that every object has some kind of a baseline of emission that creates a fluorescence. Okay. And so the key thing is if everything glows green, the key is actually the difference of how much more it glows green when the GFP is there. Okay. So that's an important thing to understand with the fold change and the induction is everything's going to have sort of like a baseline reading of fluorescence. And if this is some graph of like fluorescence. And the question is, how much does that change when you add the induction agent? Okay. So that's just another thing to know, understand about autofluorescence. That's in any time you see a GFP paper analysis, this autofluorescence concept will come up. Yeah, question, ask. Did anybody catch that? The dotted line. That might be some kind. I don't know if it's actually like statistically, like if they produce this with a mathematical calculation. My impression of looking at this is exactly what you said. Is it's probably like if they're trying to identify which clones they like, they probably kind of like drew a line somehow, and they're saying like clones above this are probably like good ones. So that's probably a hypothesis. I'm sure somewhere they say what the dotted line is. Yeah, there you go. So it is some kind of a statistical measure where they're looking for things that are above that. There you go. Very good. Okay, so we covered that. Anything else that's worth? They did do a secondary method of confirmation of GFP expression. Did anybody catch what that was? It actually was not shown in the data, but they said that they did it. Yeah, they did what they did, or they call it, they use a different word for it. They say immunoblot. So because GFP is such a characterized protein, you can just buy antibodies for GFP. And so they can do Western blots to see if they're seeing expression. But they didn't show any of that data. So they just talked about it. So that's kind of like a no-no. But they got away with that here. I'm not being too harsh on this paper. Okay, final conclusions at the end of the day. Did anybody catch in the discussion or right at the end, like, did it work? Did, like, how well did the expression work? It did, but greatly, so they did say that, like, they're saying like it was greatly affected by the promoter. Essentially, the promoter was working, the plasmid system is working. But when you say greatly, it's like, is that quantitative, and how good is greatly? So they actually do have a good line that describes very precisely how well it's working. Did anybody find that? I skipped these later figures. Did anybody find that?
See the line where they say the average level of expression.
It's close to that section. There is a point at which they say percent total protein. I'm look. Yes, you said I'm looking for that right now. Here, it's in the where? Bottom of the second paragraph in the discussion. Here's the discussion. Yeah. This. Yeah. So, wait, no. They're saying that on normal, this is actually a different organism. In Chlamydomonas, they can get ranges of 1 to 5% total protein. And here they're saying that their expression is equivalent to approximately 1.48% standard deviation of total protein. So essentially, when you make algae and you extract total protein from algae, 1% or 1.5% of all the protein in the algae is actually GFP. That's pretty decent. Like, that's pretty good. At least that's a baseline off which you could maximize or optimize or increase if you wanted to improve the system. So this is the line where they're actually telling you like how well this works. And any future experiments in optimizing the system would want to compare to this sort of baseline level. Okay. So those are kind of the main points of the paper. Are there any like major questions about it?
Like in just regular algae, the average level of GFP is around 1.48% total, and they got it from 4.2.
Did they say 4.2? Where does it say the 4.2? Yeah, right here. Yes, no, you're right. Okay, so okay, yes. So using the nitrate reductase promoter, it bumped up to 4.2. So that's what, like a fourfold increase? That's another question. Is that really that great? Fourfold increase? I don't know. The thing is, you'd have to compare this to, like, my mind is comparing this to E. coli. Like in E. coli, you could probably get a much better yield, but for algae, this might be amazing. I don't know. You'd have to kind of read a few papers, and that's why they are comparing this also to Chlamydomonas, which is another algal system. So another model organism. That's the Chlamydomonas. That's Chlamydomonas, yeah. No, that's okay. But that's my point is that in order to figure out how good or how well this is working, they try to get a baseline actual number and compare that to other systems and see where it's going. And any future studies should then compare to that baseline number to see if they can improve the system. Okay? So that's the gist. The intention of you reading this paper and taking the quiz was to hopefully be able to now apply the knowledge that you've learned from the class. And hopefully now you can read and understand scientific papers in this field. Okay? Very good.